For arranging this wonderful presentation of someone I know since last 3 decades, a very close friend of ours and a leading scholar in the field of biological sciences, a person who has played an extremely important role in the development of science and technology capacity in the developing world, including Pakistan and Bangladesh and many other countries of the world. Dr. Ahmed Abdullah Azad is a dear friend, a leading scholar, and a person of great human attributes. He has visited many countries of the OIC region, currently residing in Australia, but has a very strong linkages with many countries in the developing world and continue to strive for the excellence in science in various regions. So he has a very important role in the development of science and technology in different regions. Dr. Azad obtained his BSc Honours and MSc Biochemistry from Dhaka University, Bangladesh, and PhD in Molecular Biology from University of Toronto, 1973. After 2 years of postdoctoral training in Canada, he joined the faculty of Australian National University Research School of Biological Sciences. He then joined the Commonwealth Scientific and Industrial Research Organization, which is a premier research organization in Australia. And he is since then associated with the Division of Biomolecular Science and Biotechnology in Melbourne in 1980. And he has been leading various programs, especially in the field of molecular virology. And we all understand how important this field is in the context of COVID-19. He has promoted the position, he was promoted the position of Chief Scientific Research Scientist in 1990. His research helped in discovery and development of anti-flu drug, neuraminidase. He led the research in the discovery and development and commercialization of a recombinant subunit vaccine against a highly immunosuppressive virus, for which he was awarded the highly prestigious CSIRO Chairman Gold Medal for Exceptional Achievement. He made seminal discoveries leading to better understanding of the molecular basis of HIV/AIDS, pathogenesis, and strategies for arresting progression to AIDS. In 2000, he accepted the newly created position of the Director of Research at the University of Cape Town. This position and other appointment as board members of the South African Medical Research Council, as a member of the Council of Scientific Advisor of ICGB, which is International Center for Genetic Engineering and Biotechnology, and Trieste, Caprisa in Durban, and other very important assignments in different countries of the world, led to extensive travel and interaction in sub-Saharan African country, which promoted him to devote his time and expertise to research capacity building in Bangladesh and other technology and resource constrained countries, which he has been doing pro bono since his retirement in 2007. He is an elected fellow of different science academies, including the World Academy of Sciences and Secretary General of Islamic World Academy of Sciences, which is one of the organizations established by comstech. Dr. Ahmed Azad Khan, apart from his scientific excellence, I think the most important contribution he has made was placing young scholars in different countries of the world for research training. And that has led to development of centers of excellence in many countries of the world, and also very active research in various areas of biological sciences. He is a true mentor, a great friend of ours, and also a person who has great understanding of the science and technology impediment opportunities in the Muslim world. With this, I would request now our dear friend, Professor Dr. Ahmed Azad, to kindly deliver his lecture on a very interesting topic. Over to you, sir.
Thank you very much, Dr. Chaudhary, for this very kind introduction. As you can see from the heading, I'm trying to talk about two very new areas of therapeutics, which I think if we all work together, we'll be able to bring affordable medicine to the people in the Islamic world. And if we play our cards right, we might also make this an opportunity to create wealth. Everybody knows that we have a long way to go in the Islamic world. Of the 57 OIC member countries, not a single one is classified as economically advanced. 21 of these countries are classified as least developed countries, and these LDCs, plus many more, are still classified as scientifically lagging. So it's our duty to really see if we can bring the whole area up in science and technology. It doesn't take much to guess that economic advancement and sustainable development cannot happen without excellence in higher education and postgraduate research and the presence of a strong science and technology base. For the socio-economic development of the Islamic world as a whole, it is important that we think about the LDCs and technology poor countries and take them along on the journey with us. We all like to do everything that is interesting in the world. And maybe in one day, the Islamic world will have the capacity to do those sort of things. But for the moment, it is not easy for any country to be competitive in all areas of research because of lack of resources, trained manpower, and infrastructure needed. It's better that we do fewer things, but do them better at a world standard. And so we need to focus in areas of common interest, in areas where we have existing strength, and development priorities of the Islamic countries and the Islamic world as a whole. And do this through establishment of multidisciplinary and multi-institutional collaborations between different groups in different Islamic countries who possess complementary expertise and facilities. And I think there's no better organization than Comstech to lead us in this area. One area of priority research and common interests that can involve all Islamic countries, no matter from which economic state they are in this research is the production of biotech medicines, especially the medicines from the Biota, for the affordable healthcare and wealth creation in this part of the world. The two areas of therapeutic research that I'll be talking about is one is the development of affordable biosimilar medicines, which is not based on basic research to start off with, but copying something, but it's copying something which is so complicated and involved that we have to start with that to make some inroads and make money. I mean, don't forget that Japan and China and countries like that started off with copying things on a small scale, and now look where they are. So there's no shame in copying things which are important for our part of the world. And the next one that I'll be talking about, and that I've talked with Dr. Chaudhary quite often, is the rational bioprospecting of our natural resources for the discovery of new drugs. Because this is the one area where there's a huge opportunity for all countries in the Islamic world to get involved. And there are immense prospects for intellectual property development in this area. Now, before I go on to the rest of the talk, I have to really define a few terms. I'm not going to talk about generics, but I'm just going to use it as a reference point of where we are at the moment. These generics are exact copies of small molecule drugs which are patented, and they can be easily copied. And because they are exact copies, there's no need for clinical trials. And this is the bread and butter for most of the companies in the Islamic world. But the problem is that even there, our own countries don't seem to be making the active chemicals needed for these drugs, and we are importing them from other countries and repackaging them, and the so-called fill and finish technology. But still, it's producing drugs which are of very high standard and are very useful and are quite affordable. But in recent times, what has happened is that the multinational companies which used to make the blockbuster small drugs have moved away from there because they are finding that a new class of drugs which are based on proteins and peptides called biologics are much more important, and because they are very efficacious and very specific. And what they do is they take things like enzymes and proteins, like hormones and growth factors and things like that, and make recombinant copies of these. And this was the first generation of the biologics. And then they went on to make monoclonal antibodies, which very specifically hone on the target molecular targets for therapeutics. And they started off with using hybridomal technology. I'll talk about it soon. And then there is a new class of molecules, which is called the Fc fusion proteins, where they take the very conserved part of the monoclonal antibodies and add on the active drugs, protein drugs, or peptide drugs onto it. The problem with these medicines, they are extremely efficacious. They don't have side effects, and they are very good, but they cost in excess of $50,000 person per year. This is the minimum. They cost even more. In countries like Australia and others, the government subsidizes some of them, so we can buy it, but it's very difficult in the developing world. So the major interest now is going on to, in our part of the world, is to biosimilars, which are cheaper versions of the biologics, which are also produced by recombinant DNA technology. They are called biosimilars because they have the same therapeutic properties as the biologic medicines, but structurally they are similar to, not identical to the original biologics. And because of that reason, there are clinical trials needed, and that brings in complexity, and we have to really answer these questions, how we go about doing it. The most the biosimilars sold by pharmaceutical companies in the Islamic countries, like the generics, are repackaged active chemical ingredients imported from the rapidly advancing countries. And a small number of Islamic countries have locally produced the active chemical ingredients of the biosimilars, especially for the first generation biologics, insulin, interferon, erythropoietin, etc., from seed clones obtained from outside, and specifically from the International Center for Genetic Engineering and Biotechnology. And I'm happy that I could help in this respect to get these sort of things to countries like Bangladesh and Pakistan and other countries. The real challenge is in producing our own molecular clones for biosimilars of this new generation and very expensive monoclonal antibody Fc fusion proteins that are still under patent. Now you might say, why do you go after things which are under patent? We can't do it, but luckily the LDCs, and including the LDCs in the Islamic world, are not bound by the patent restrictions, and we can legally do this work, provided some of the LDCs have the required competencies. Besides producing the affordable high drugs, this is also an excellent opportunity for wealth creation for the countries who work together to make these drugs. The global market for biosimilars is definitely much smaller than that for the biologics, the original biologics, but it's still not insignificant. In 2014, when it was new, the world market was about 2.5 billion, and it surpassed more than 20 billion in 2020, and is expected to exceed 69 billion in 2025. And even if you get 10% of this for ourselves, you can imagine the amount of money we can make and save. Now, this is, I think, a golden opportunity for us to make affordable monoclonal antibody and Fc fusion proteins for our own Islamic countries and other developing countries. The monoclonal antibody and Fc fusion biologics are extremely expensive, as I said to you before. So they are not affordable by the common man on the street or to the poorer countries. And so there is a huge demand for producing cheaper biosimilars. As I said before, the LDCs are exempted from patent restrictions, so they can legally copy any patented drug on the market to produce and market them at a cheaper rate. This is an excellent opportunity for the LDCs, provided they have the required competencies and if they can work with the more advanced Islamic countries to make use of the WTO's waiver and take advantage of the low labor costs to produce affordable biosimilars for the Islamic and developing world. I think the Islamic world as a whole should make use of this unique opportunity as this provides a huge competitive advantage over likely competitors like China, India, Korea, and Brazil, who cannot touch these patented drugs till they come out of protection. And among LDCs in the Islamic countries, I'm sure there are more, but Bangladesh is one country that has built up most of the required competencies, but it still requires a lot of help from more advanced Islamic countries. Now I'll just go and explain what this is. This is a linear representation of an antibody molecule where you have a light chain and a heavy chain, and there's a light chain which are joined together by disulfide linkages. And if you can make this one copy, then a lot of those, then another copy will come and automatically join together to the disulfide linkages to give you this Y-shaped molecule. And that's what we start trying to do in the very beginning. It's quite a complicated task because it's about 150 to 180 kilodaltons in size. So you can imagine that it's not a small thing to do. I'll first talk about a few of the TNF inhibitors. TNF stands for tumor necrosis factor. It's a very important protein in the human body. It's important for the regulation of cell growth and tissue development and a myriad of other things in the body. But in some disease conditions such as arthritis, different types of arthritis, Crohn's disease, and other inflammatory diseases, what happens is that the TNF, instead of being a good agent, becomes a very bad thing for the body and causes a lot of pain and suffering. So some of the first monoclonal antibodies that were prepared against TNF were these monoclonal antibodies. And the first one that was prepared was this one here, which is a chimeric monoclonal antibody produced by hybridoma technology. What they did here, and this is the sort of work that got Cesar Milstein and others the Nobel Prize quite a few years back, was to inject, say for example, I'm just using TNF as an example, the TNF into mice, and the mice make antibodies in their B cells. And what they do is they take out the B cells, individual B cells from the spleen of mice, and then try and fuse a single cell, B cell, with a myeloma cell, which is a tumor cell, human, which doesn't die because it's immortal. It's a cancer cell, which is immortal. And if you join the two together, it becomes a bigger cell, and it's called a hybridoma cell. And what the hybridoma cell does is produces a chimeric monoclonal antibody, where part of it is the human antibody, and this green part here is the mouse regions which join down, and this is the region which are specific for binding to the TNF. This molecule works well, but the problem is that if you want to repeatedly use it, it's not working, because the human body recognizes the mouse sequences. It rejects this when you have it, when you try to inject it the second or third time. So it doesn't work for long. Then in those days when recombinant DNA was not that established, what they did was try and very manually try and remove the much of the mouse sequences from this region and replace them with human sequences. It worked better than this in being a bit more, working for a bit longer, but still they were rejected because the human body recognizes these as foreign proteins and rejects it. And nowadays what they do is make the whole molecule as a human monoclonal antibody, and it really works very well. And I'll go on to tell you about this, how they do it. You don't have to synthesize the whole thing. What they do is, this is again linearly shown. You make a copy of this heavy chain, and then you add a linker there, and you make it here. And this is done through the gene for this protein, and then you put it in a vector and produce the protein in recombinant protein, and you end up with one molecule like this, and 100 other molecules is similar, and each one then joins to the surrounding one and makes an antibody. And so this is a totally human monoclonal antibody. I'll not talk about the FC fusion proteins. And here, look at the molecule. I mean, this is, previous one is a linear depiction, but in reality, this is also not real. But the antibody molecule actually has very specific domains. This is the heavy chain, and from here onto this part is completely conserved. It's constant domain, and this in the light chain, this is also very similar to that. These two are joined by the disulfide bridges, as are these molecules, these domains joined to each other through disulfide bridges. But if you want to make add on a therapeutic protein, like let's say, for example, there's a receptor which is very good as a therapeutic. So what you do is you remove this completely and you replace it with this molecule here, and you end up with molecule like this. And this is a FC fusion protein, because you have the FC region, this is the conserved region, and you have the drug which is joined on. Now this one is a similar thing. I'm just trying to show the sort of things we are trying to do. We don't have to make the whole molecule. What we need to do is we make a gene corresponding to the CH3 domain, the CH2 domain, and go up to here and conserve the disulfide bonds. And we use the gene for linkers, amino acids like serine or lysine, and join it onto the therapeutic protein gene, the one that we want. So the gene for the therapeutic protein is joined onto the gene for the constant region. And when it is cloned and expressed, then this is the molecule it produces, and there are lots and lots of copies. So each molecule will be joined another like this, and something that will look like that. So because of this H3, is a peptide linker, the therapeutic protein, which can be very big or small, it can move about to seek the target and bind to it to neutralize it. So this is basically what we want to do. Now, the most important part, or the most difficult part, is the upstream processing for reverse engineering. And in some cases, you can get ideas about the sequence of a new drug on the market, new therapeutic biologic from the patent literature, but most often you will not. And what you can do is you can just buy a few tablets or capsules of this new biologic, and use a tandem mass spectrometer to sequence the N-terminal protein. Now you might say, well, it's cheating. It's you are stealing something. It's not really that. It's because the uh, the these developed countries in the Islamic world and the developing world are not constrained by any patents, so they are allowed to copy and produce anything that's available in the market. Now, usually you know the dogma that you go from DNA to RNA to protein. Here we're going the reverse direction, and that's why we're calling it reverse engineering. So once you know the amino acid sequence of the N-terminal part of that new biologic, it stops where the heavy chain of the monoclonal antibody, which is preserved, comes in. We just get the sequence N-terminal to that. And from this amino acid sequence, we design the equivalent gene and make sure that when we have this gene that we use human codons. Because this is for two reasons. The codons, which are triplets of nucleotides, some might have, there might be multiple codons for the same amino acid, but some are specific for bacteria, some are specific for the lower animals, and some are specific for humans and apes and things like that. So I use the human codons, both for the fact that it's not going to be rejected, and also because of the fact that when you produce it, you'll have higher yields. Now, it's very important to chemically synthesize our own gene because it can be done commercially. It's not, doesn't cost an arm and a leg. But if you send it out to be synthesized outside, then the cat is out of the bag. So it's best to synthesize it in-house or with the help of another country which can make this available to us. And now what we also have is a fusion, has an expression vector. I won't go into details about it, in which the constant FC region of the IgG is already built in. So the gene that we have synthesized goes on N-terminal to that, or you can say the in case of nucleic acid, the carboxy terminal adds on to the N-terminal of the FC region. And so we have one gene, and which when you take it through the recombinant DNA process and express it, then you end up with this sort of a molecule, which I just showed you before. Now, we have to then do small-scale fermentation in Chinese hamster ovary cells for expression of the biosimilar to ascertain its integrity and biological activity. Now, what happens next is that in the past, we used to spend months and years to get a high-expressing clone, which would be high enough for commercial purposes, because not producing in high enough amounts is really not commercially viable. But once you do it manually, like we have been doing for many, many years, it can take months or years to get even one. So what we have to take advantage of is a semi-automated clone selector, which has a built-in fax and a cell sorter. And with that, you can select single cells from the expression library you have produced, and the single cells can then be separated and then grown with under very different conditions, 24 to 48 different permutations and combinations, and then you can check it very quickly within a week to see which one of these clones is giving you high enough activity or not. And suppose it might take one cycle or more than one cycle to get it, but usually you do get something which is very good. And something like 3.5 to 5 grams per liter of the very expensive medicines, is a good bet that it will be commercially viable. Now, in terms of this automated clone selector, in Bangladesh, there is only one in a pharmaceutical company, but it's not available to anyone else. and it's quite expensive. It's about $500,000, just as the tandem mass spectrometer, but it might be there at the ICCPS. I don't know. I'll try to find out after that thing. Okay. Now the next step, which is also a huge task, and the pharmaceutical companies who set this up have to spend hundreds of millions of dollars to do it. The first step is to go stepwise to produce the biosimilar protein by batch fermentation in a 2 to 5 liter fermenter, and then ascertain its structure and biological function, activity. If it works, then you go on to a bigger fermenter, and then you to get the conditions right and produce enough of it for preclinical animal studies. And if they look all right, then you go on to the large-scale fermentation and purification of the biosimilar under GMP conditions. The one for the preclinical studies doesn't read GMP conditions, but this is GMP conditions, and this is a very, very expensive process. But I'm glad to say that there is at least one company that I've been working with, and there are probably more now, which have large-scale fermentation and purification facilities which are second to none in the world. And many of you who are fellows of the Islamic World Academy of Sciences had attended the conference in Dhaka in 2013, had the opportunity to go and visit the plant which produces this. So this is there, but there's no guarantee that this company will work with us. Now, I'll be talking about preclinical animal studies and human clinical trials and regulatory approval again and again, but so that I don't have to repeat it, I'll just spare you please. For the preclinical animal studies, a lot of Islamic countries have the facilities for looking at their safety and bioavailability and things like that. But sometimes you have to work with anti-infectives, in which case you have to have challenge studies. For example, you have found a drug or a vaccine against the coronavirus, the COVID-19 virus. Now, one of the ways to do it, and I think that should be done, is to have a facility which is specific pathogen-free, and it's also have high containment like BCL3, which is very expensive, so that you can actually treat these animals beforehand with the vaccine or the drug, and then challenge them with the live virus to see if these animals can actually remain uninfected and get rid of the virus. But you want to do it under conditions that they don't escape from the laboratory. And it is important that we have this facility. It's not in every country, but at least regionally, or at least in one place in this Islamic world. Regarding human clinical trials, I can tell you that it's very expensive. The first one I was involved in was during the production of the anti-influenza drug, which was the first structure-based drug in the world, actually. And we went to Glaxo, and they had to spend $500 million to do it. I'm sure we can do it much cheaper than that if we control it ourselves, because I have been involved in providing funding and expertise to the groups who are working in South Africa and the rest of Africa, and it can be done under our own conditions much more cheaply and just as well. But we have to have the will to do it. Now, this is another thing, the regulatory approval for releasing it to the market. I find it is very difficult in the sense that in some Islamic countries, it's a simple question of common sense where a senior scientist can go and talk to a friend who's a minister of health or some other minister, and they can convince them that they are not compromising safety and efficacy, and they get approval. And then there are lots of other countries which have regulatory guidelines that are stricter than what you have in FDA and the EUMA. And in fact, they can combine the most difficult things from the two and put it together to probably prove that we are better than them. But what they don't realize is that these guidelines that have been produced by the FDA and by the EU MA are actually there to stop us from going to their markets. And sometimes they sit down with their own people and decide on how to do it, and this is what we have to do. But we have to have a simple and uniform guidelines for the Islamic world. In terms of manufacture and marketing, I think we are pretty well established, because I can tell you that in case of Bangladesh, we produce 97% of all the medicines we need locally, and we export very high quality drugs to more than 130 countries, including the USA, Canada, and the European countries. And luckily, we also have a very good market in sub-Saharan Africa and in the least developed countries, where our drugs are affordable and they're very well appreciated. And there may be other countries we can do it. So this area is quite well established. So the question is, should Islamic world produce these new generation biosimilars? Now, as I said to you, the biologics extremely expensive and beyond our reach. And so there's a very high demand for the cheaper monoclonal antibody type biosimilars and the FC fusion type biosimilars. I think the production cost of these proteins would be greatly reduced if the biosimilars were based on patented biologics, and if we made it from the seed clones developed within the Islamic world world. And we already know that we are allowed to do it by the WTO and the least developed countries. And this provides us a huge competitive advantage over countries like China, Korea, and India, who can only work on out-of-patent biologics. But we cannot hold on to this decision, because the time of the patent, where other countries are wanting the patent to come off very quickly, we are trying to make use of the time of the patent to make all the money we can make and all the drugs we can make before we have to compete with countries like China, Korea, and India. And so we cannot delay thinking about it. We have to get working. Now, the question is there any LDC in the Islamic world which has at least some of the essential competencies? And how are there countries in the other scientifically advanced countries who can help? I think that there are yes to both of those questions. And I'll just talk about the critical requirements. The upstream processing, as I told you, the reverse engineering part, it must be undertaken in an LDC, and in Bangladesh and probably other LDCs I'm not aware of, but there might be. We are quite capable of doing this, and we have set up a team which can do it. But what we really lack is the semi-automated clone selector. And while there is a moldy tuft of MS in one of the pharmaceutical companies in Bangladesh, they are using it 24/7, so we can't get a hold of it. CSIRO has the LCMS, but I don't know whether there's an LCMS. And we don't need it every day, but initially and for ascertaining what we have produced, it's very good to be able to sequence the protein that is produced. And if there are some advanced countries, such as Pakistan, the ICCBS, if they have it, we'll be very grateful. And the gene synthesizer is not very expensive, and it might be good to have some of these in different parts of the Islamic world, so that we can make our own genes instead of sending it out to be synthesized and then letting the cat out of the bag every time. In terms of large-scale fermentation and downstream processing, I've said that there's one and probably more drug companies in Bangladesh who have actually produced biosimilars from molecular clones. What they have not done is the upstream research I was talking about. And they may or may not work with us, but we have to find the companies that can do it. But I think that capability is animal studies or the clinical trials with the regulatory approval and the manufacturing and marketing. The last one is very well, we are already very well placed. I'll now move on to a second type of research, which is actually carried out in many of the Islamic countries. Here we're trying to combine traditional knowledge with modern science, and that's why I'm calling it rational bioprospecting. Not that what was done before is not rational, it's just that we are introducing something new here, trying to introduce. I've talked to Professor Chaudhary before. I think he agrees, and we can start on it. Let's look at the strength of why we can do this. We have a huge knowledge base, indigenous knowledge of traditional medicines, and every Islamic country has lots of regional flora and fauna and the biodiversity hotspots, which can really help us get us started. And in fact, ethnopharmacologists, ethnobiologists have gone to the traditional healers and used the flora and fauna, especially in the biodiversity hotspots, to look at the different plants and publish this, and they have published it. It's one of the highest areas of publication in the Islamic world. I haven't found too many places where they have got extract libraries from many different plants, but it can be done. And then we have the people who, chemists, the medicinal chemists, who have done a wonderful job of taking the medicinal plants and getting out the secondary metabolites and then finding their structures and trying to find what their functions are. And this is also highly published and one of the highest published areas in the Islamic world. And I'm sure that they must also, at least in the ICBS, I know they keep libraries of these isolated secondary metabolites. So these plant extract libraries and the secondary metabolite libraries are actually gold mines of undiscovered novel drugs. As I said, there's very extensive and impressive research publications, but unfortunately, these have rarely been translated into patents and new medicines. There have been instances where they've been done, but it could be much more. And one of the reasons for that is that the multidisciplinary collaborations are still new in our country. Like the ethnobiologists and the medicinal chemists should be working together, but I don't see that, at least not in my country. Then there's a huge opportunity for generation and utilization of IP and patents. This is not a culture in the academic laboratories in home. And even if you did patent, there's no money to continue with the patents or to protect them. And so a lot of patenting that's done in our country, people think that's the end. If you patent, you've done everything, and the companies will come running to you. But that's a fallacy. Actually, this is the start of the interaction with drug companies, and to attract them to this thing. But unfortunately, at least in my experience, the interaction and partnership of the academic research laboratories with the drug companies is very negligible. But it is extremely important that it happens. So why I'm calling it rational bioprospecting of the biota. Now, what is different from what has been done before is that we want to start with the disease-specific molecular target and then make assay based on that. And that assay, if you use to screen the natural product and isolated chemical libraries, have a much higher chance of giving us good leads, because these secondary metabolites in plants are unique structures, and it is well known that many of them have produced real drugs in the past, and even now it's working in the herbal medicines and also the other medicines that have come out of the medicinal chemistry labs. So the other thing is that if you use such a very specific molecular target-based bioassay, then the lead and the candidate drugs that come out of it will have a very high specificity with insignificant side effects, but so you have already done a lot of the work to start off with. And as I've said before, there's ample opportunities for IP generation, which can be used for technology transfer and partnerships with industry. And there will be high demand for molecules like that, because unlike the biologics and biosimilars, the small molecule drugs are not being developed and the pipeline is almost drying up. So there's very high potential for multidisciplinary and multi-institutional collaborations, which involve scientists from PLDCs and scientific lagging countries also. Now I'll just recap the discovery and development pathways. What I term as discovery research is 4 things here. One is the identification, validation of disease-specific molecular targets. This has not been used for natural product-based drug discovery in the past, because the molecular biologists have never been part of it. But what the molecular biologists don't realize that if they make, and they publish very nice papers, but they don't go on to develop a molecular target-based bioassay because they haven't had a need to. But if the molecular biologists and the people who are doing the initial looking at the ethnopharmacology and the medicinal chemists were all working together, then they would know that such a bioassay would be extremely useful in the identification of new lead compounds by screening the natural product and the libraries with these highly specific molecular target-based assays. And this will be something which is a completely new way of looking at things. Not that nothing has happened, but this is a new way because you'll be getting very specific leads. And there are at least 2 or 3 different stages where there's opportunity for new IP. Now in the development research also, you can use this highly specific bioassay for bioassay-directed lead purification characterization, which is already being done in different labs, but they often do it with bioassays available commercially or off the shelf. But here it is very specific, it is patented, and so whatever you use here, it belongs to you, and if you find something, purify something, then it is your compound that you're doing, and there's new opportunities for IP. Now, what we would like to do with the leads is optimization of the lead molecules into possible candidates, and it needs a lot of chemistry. Professor Chaudhary can tell you about that using combinatorial chemistry and analog chemistry. And new things like structural biology and, of course, computational chemistry. In structural biology, I'll just tell you a story of a friend of mine in New Delhi. And he was struggling to crystallize a very important antigen for malaria. And he couldn't do it, and he heard from faith healers that such and such plant has got something in it which works very well for malaria. And he actually mixed his very purified protein with the extract from that plant, and he recovered his protein and purified it with a molecule bound to it, a natural product molecule bound to it, and lo and behold, it gave a 3D structure very quickly. So you'll be amazed where natural product can lead you, and there's no need for us to think that that's ancient and something else is new. We can always work on the same things and get new ways out of it. And of course, here we cannot select without the predictive and animal studies. And as I said, we need very uniform and regulatory affairs guidelines. And manufacturing and GM conditions is already there, and we're very good at marketing. So because it's the time of COVID-19, I'll just give you an example of a molecular target. This is not something that somebody has done or I read somewhere. It's just that comes to my mind that can be done. Maybe somebody is doing it, but I don't think anybody is involved using natural products for this. You know, the new vaccine that's supposed to be 90% effective for COVID, it targets this spike protein, and especially the receptor binding domain of the spike protein. And this protein actually binds and attaches to the human cells through a surface protein, a surface human receptor called ACE2, which stands for, what's that? It's a very important protein for cardiac angiotensin converting enzyme. It's very important for control of cardiac activity in humans. So the target bioassay is developing an assay where you're observing the in vitro binding of the virus ribosome binding domain to the human ACE2. And there are many different ways how it can be done. I would do it in a 96-well microtiter plate, and we can talk about it if anybody is interested. And how do you find the therapeutic leads? If these two are binding, and if you can find a natural product extract which stops the binding, then you look for what is that is stopping the binding, and that product could well be a future inhibitor of this binding, which prevents or disrupts the binding of the viral segment to the human protein. But at the same time, you have to do when you have done more work, you have to make sure that whatever you have discovered, the new lead or the candidate drug, does not disrupt the biological activity of the ACE2 enzyme, because it's extremely important for humans. There's another example I can give you. The RNA of the SARS-CoV-2 virus, the one that caused COVID-19, is copied by a non-structural protein of the virus, which is an RNA-dependent RNA polymerase. So what it does is takes the RNA polymerase and makes multiple copies of it, which can get packaged up inside new viral proteins. So if you can stop the replication of this viral RNA, then you will stop the replication of the virus. So the target bioassay is that you use this RNA-dependent RNA polymerase and observe viral RNA synthesis, which is the same as that for the virus. And that's where the sequencing becomes very, very important. So DNA and RNA sequencing is also very important. So what would the inhibitor in a natural product do? It would specifically inhibit the production of this virus, which means that this viral protein, viral enzyme cannot produce viral RNA anymore, and you have a drug. But again, in this case, you have to make sure that the lead compound that you have discovered does not inhibit any human RNA or DNA polymerases. That's also very important. So what's the take-home message? The take-home message is that there is unlimited potential in the Islamic world for production of biotech-based pharmaceuticals for ourselves and for the rest of the developing world markets. But this will not happen without political will and without coordination at a high level and adequate funding. If a comstech could convince the governments of the Islamic world that the biggest investment they can make in their own future, in the future of the countries, is to spend the adequate amount of money in education, research, and innovation, like 20% in education, and at least 2% of the national GDP on research. If you don't do it, nothing is going to happen. And as I said, there's enormous potential for academia industry collaborations, and we have to work towards getting this, making this happen in some way. But one of the biggest problems we have in our part of the world is there's a huge developmental gap between discovery research, where molecular biologists and others go and, or the medicine chemists go and discover whatever they publish, and the pharmaceutical companies who get their chemical ingredients from overseas and repackage and sell them. So what is really missing is the developmental research that comes between discovery research and commercialization. And we cannot reach our full potential till we can bridge this R&D chasm. I could see you a show of large number of slides, but I won't bother. And then go on to the last slide, where I'm just making some suggestions of how comstech can play an enabling role in research capacity development in molecular biosciences in the Islamic world. So if you're going to have a very good multinational and multi what you call different using different technologies, then you need to have administrative policy and logistical support. And there are some ways where comstech can really help. One of the things that we might start thinking about is to establish a trust foundation like the Wellcome Trust in the UK or the Rockefeller Foundation in the US to adequately fund collaborative research and development in areas of common interest and highest national and Islamic world priorities, funds enough PhD, postdoctoral training and visiting fellowships, and also provide the essential equipment and pay for the human clinical trials. If we don't have that sort of money, we can do all the research we want, but I don't think we'll get anywhere. What we also need, and I'm talking about molecular biosciences, but it could be any other area, establish a world-class molecular biosciences research center, which will not only work for drug discovery and development, but it will work for food production, for agriculture, for environmental biosciences, for energy production. These same technologies will be useful there too, with state-of-the-art equipment and technologies that most Islamic countries cannot afford to buy and operate individually. Some countries have done that and come a long way, and maybe because of individual leadership also, like in Pakistan. The center could be an educational and training hub for young researchers in our part of the world, especially those from least developed countries and scientifically lagging countries. And this centrally located research center could be modeled on the European Molecular Biology Laboratories in Heidelberg or the International Center for Theoretical Physics in Trieste, which was established by our own Professor Abdullah. And finally, I think that the headquarters in Islamabad can set up a technology transfer office as a one-stop, it might already be there, as a one-stop advisory and support hub for issues related to IP, regulatory affairs, and commercialization with uniform and harmonized guidelines. For regulatory affairs, comstech would follow the example of the European Molecular Agency, Medical Agency, which covers all European nations, and we need something like that to cover all countries in the Islamic world. I think I'll stop there for the moment and take your questions. Thank you for listening to me. Thank you very much.
Thank you, Professor Azad. The floor is ready for the questions. So if you have any question, kindly.
I have a question for Professor Azad, if I may.
Yes, please.
Can you hear me?
Yes, I can hear you.
Please go ahead with your question.
Hello, Professor Azad.
Hello,
I'm so
friend.
glad to see you.
I haven't seen you for a long time.
We haven't seen each other for a while, but I'm so glad to see you, and I'm very glad that you have once again exposed a roadmap for Islamic countries for the world as to what can be done in the domain of drug discovery and development to meet our immediate health needs and really contribute to the global effort to stand up to the COVID-19. I think if Islamic countries had listened to what you proposed 15 years ago in terms of establishing the infrastructure for drug discovery and development, then today I think we would be in a much better position to stand up and address the COVID-19 virus. Sadly, like many other instances, we never miss an opportunity to miss an opportunity. And this is what happened when you started calling for this 15 or even 20 years ago. I think what the COVID-19 situation has exposed is that the Islamic world in particular, the 57 countries of the OIC, are not at all ready to do something in terms of the vaccine development or the cure development of the COVID-19. Really, our efforts have not been fruitful. We have let most of our scientists have let our countries down in terms of standing up to the COVID-19. So I think I would ask you to briefly elaborate on your last two slides in terms of the roadmap that we have to have in Islamic countries to really be able to also develop our own vaccine to stand up to the COVID-19. Also develop our own cure, utilizing our own resources. As I said, I very much subscribe to the roadmap that you have outlined. But I'd like to hear a little bit more about how we can collectively and even individually as countries, what we can do immediately to help develop the vaccine or the cure for the COVID-19, and in general, really stand up to the global pandemic. Thank you.
Thank you very much. I think I agree with everything you say. But I think in terms of getting a vaccine produced in the Islamic countries, we might have missed the boat. I can tell you one thing, that even if we wanted to make the vaccine, there's not a single country that I know of where they would be able to do the trials for looking at the live virus, because you have to do these infection studies where you use your vaccine or drugs in the animal models and then challenge them with the virus, and we don't have one. And that's why I keep on saying for 20 years that we need to have a facility, a central facility where everybody has contributed to set up, so that we are not constrained in doing experiments like that. For example, there's a group in Bangladesh who came a month back saying that they have produced a vaccine, but unfortunately, the vaccine they were doing is very similar to the one that is supposed to be 90% effective. But what they think, that they really didn't know, is that producing a vaccine doesn't mean making antibodies. Any protein injected into any animal will produce an antibody. What you really need is to produce antibodies that neutralize the virus. And in order to neutralize the virus or to do the challenge studies, you have to have facilities where you are able to use it. They have their BCL3 facilities, specific pathogen-free facilities, which are not easy to what should I say, produce everywhere. So I've been saying, as you know, for 20 years that we should have facility like that, centrally located, not all everything in one country, but in a number of countries, because we keep on talking about science in this country, but itself, it is only, it doesn't get anywhere. We do fantastic work in ICCPS and Karachi, but in terms of the things I'm talking about, there are not too many places. I think they have a lot of places, they have the potential, but I don't think that they click together. The culture of working together towards a common goal doesn't work. The Islamic Development Bank, for example, gives out the small amounts of token money every year to one or two projects. And this doesn't really take us very far. And the second thing is that many of our brethren who are floating on oil and have money to burn do not contribute to the general fund for making something, because they have the money to buy the technology from overseas. But what they don't realize that when they buy technology, they don't develop their own capacity. So there has to be a change in the way we think and then the mindset.
Do we have any other questions?
Doesn't look like Hazima.
Okay.
Can I?
Yes, please, madam, please ask your question.
Yeah. First of all, glad to thanks for your this presentation. My name is Sri Fatmawati from ITS Indonesia. I'm also working on isolation and identification. So basically, I'm chemist, so I'm working with the isolated compounds. So actually, I need your advice, maybe a kind of advice. Regarding our, you know that right now we all of us in the same condition, yeah, in the pandemic. In my lab, we also working with the Indonesian traditional plants, and we try to isolate some compounds that, hopefully that one of them can be candidate for the drug, or maybe in the near future we will use it for the drug for COVID. So do you have any suggestion regarding this pandemic with our isolated compounds, or what should we do for example, the bioassay or kind of in vitro assay that maybe useful for us to select which compound that's suitable for COVID-19. So
yeah, I think you are in the same boat as everybody else in the Islamic world. You will probably go and isolate the compounds and you'll publish. But to find out if they have got any function, you have to have the assays. But where are the assays? I mean, the thing is that you can buy an assay of commercially, but they're not going to be specific. And what are the assays that are sold commercially for COVID? I don't know if there are any. So I was just talking about two which came to my mind to give an example of what you need to do. But if everybody like you had their libraries and we all decided that, okay, we'll work together, but let's first find somebody who has got an assay. And if we use that assay and use our molecules to see if anything works, then we go forward and we share the intellectual property. Both the people who have developed the assay and the people who have got the libraries are sharing in the discovery. But unless you have these specific assays, you're not going to get anywhere. Now what most people do is they find the products and then they try and find the function for it. It's like the actors who try and find a role for themselves on stage. So you have to actually combine things together, and I can't blame you for anything. You're doing the right thing, but maybe Comstech will convene a meeting where we try and invite molecular biologists and cell biologists and other people who are not in the loop to come together and see if we can build some assays, because without the assays, both the in vitro assays and the infection assays, we can't go anywhere. And it has to be a centrally done thing. I'm sorry,
Okay.
I can't give you any assays.
Yeah, thank you so much. Thank you.
I just gave you 2 examples. If you must have the people in biochemistry and computational chemistry and in computational biology in your university, tell them to make the sort of things I'm talking about. Maybe then you can test your products.
Okay, thank you so much.
Welcome.
Dr. Azad, we have another question from Dr. Samina Ijaz in the comment section. She's asking, can we have a common biobank for the Islamic world?
I think that would be a fantastic thing to do. What we can actually do is keep it together, but in a coded form where only the people who have supplied it know exactly which plant or thing is, because people are worried that if they give it, then they lose it. It's not that, but first of all, we need to know which are the common ones and which are ones, and the unique ones are the ones where you should probably try and number it in such a way that only the lab supplying it knows what they are. So if there is there's a lead compound that comes up, then you can go back and see what was the plant that had that lead compound, and then you can come to an understanding. But having a bank for these extracts, for both the extracts of plants and also the chemical libraries in a standardized aqueous form would be a very good idea. And that might be one of the things that is done. While at the same time, we look at what research is being done in the Islamic world, or by expatriate people who are originating from the Islamic world, who have done the research which tells them how the molecular basis of pathogenesis of a disease, and they haven't thought about making a bioassay. But if they help us making a bioassay, they can get involved in working with scientists in the Islamic world. But this is a good idea. I think it would be a good idea to have a central bank for keeping all the plant extract libraries and the chemical synthesized libraries, but under the control of really under the control of the people who give it till something is found.
Thank you, Dr. Azad, for a very nice explanation. Do we have any other question? So in case we do not have any question, thank you very much, sir. It was a pleasure and a delight to hear from you. And I'm very, very thankful for your presentation. And you can see the tremendous feedback that we have got from our participants for your presentation. Thank you to our participants. Thank you very much for joining us today. Each one of you will be getting any certificate for attending this lecture. We hope to have you in our upcoming events as well. Thank you very much, sir, again for your time. And it was wonderful hearing from you.
Most welcome.
Okay. Have a nice day, everyone. And with this, bye-bye.
Bye-bye.